So recently, in fact within the last week, we received this machine. It's a PCR machine, and you're probably familiar with PCR nowadays because a lot of people would have had it for your COVID test. So what PCR stands for is Polymerase Chain reaction. And what it literally does, this machine, it melts your DNA strands. Your DNA strands are double-stranded, it melts them, and then it extends each of those strands and makes a copy. So in that first cycle, it doubles it. So you go from 1 copy to 2, then to 4, then to 8, then to 16, then to 32, and so on and so on. So what it allows you to do, it allows you to detect very, very small quantities or very small amounts of DNA and actually amplify them to a level that you can detect in this machine. So what it allows us to do, it allows us to do a whole range of things. It allows us, for example, to look at pathogen load in the soil, or it can look for the level of pathogen in our compost. And so one of the things that we want to do initially is to actually start following some of these things within our compost. Our compost undergoes a set of roles and cycles, and during each of those, some of those pathogens that are potentially present will get killed by the heat. And so what we can do is we can actually follow that and look at the numbers as they reduce, and this machine is the ideal way of doing that because it's very, very accurate, it's very, very quick, and it's very, very precise. So that's one of the applications. The other applications are obviously for people that potentially want to look at soil samples, or even, let's say you've had a chicken shed cleaned out and you want to know has it been cleaned out properly. This machine can tell you whether there's any residual pathogens in your shed. And it really works by using these trays that you can see here. Each of these trays can do 96 different samples, so we can either look at 96 different organisms or we can look at 96 different genes. And this machine actually has the ability to look at each of those samples several times. So it means that this 96 wells can in fact be upgraded to a much higher standard. And really this is the cutting edge currently of how we can look at what's present in the soil without going through the laborious steps of isolating individual things and so on. And the other thing that we want to use it for in the future is to actually start looking at functionality. So let's say we want to look for a gene that is responsible for plant growth factor. The current way of doing that is quite laborious. We have to do a lot of biochemical testing. With this machine, we can take individual isolates and look, do they have the gene in the genome that can actually produce the compound we're after? And if they do, then those ones are worth spending the time doing a biochemical test on. And the rest that don't have the ability, you wouldn't screen them. It's really a waste of time. So it allows us to advance a lot of our research quite significantly as well.